Everybody would like to introduce Jack Gilbert. I would love to present his bio before this, but I can't actually see it. So we'll move straight to your talk, Jack, if that's okay.
It's my pleasure. Thank you, Darren. Yeah, I'm happy to give a very brief introduction of myself. I'm strangely a professor of pediatrics and oceanography at the University of California, San Diego, which doesn't make a lot of sense. Before this, I was a professor of surgery at University of Chicago. Obviously, I'm English by origin, American by nationality now. But my research is focused on microbes in any environment. I worked down in Plymouth before moving to the United States about 11, 12 years ago, where I explored the microbiology of ships and oceans and sediments, etc. And now we work in everything from agricultural activity to air and soils and oceans and rivers and plants, and a lot of human-based microbiome research. Predominantly in children, and hence the Pediatrics Association, but in many other environments. And I got into the field of the built environment around 10 years ago, a little bit more than that, when a program officer for the Sloan Foundation, Paula Olszewski, I gave her a lift to the airport or to a hotel, and she was building up this research theme for the Alfred P. Sloan Foundation. And I suggested, well, it'd be very interesting to find out what was going on in the microbiome of people's homes, wouldn't it? That'd be very cool. And she goes, well, send me an idea, and I'll see if I can find some funding for it. And so I got $125,000 to look at the homes, people's different home environments, and I'll talk briefly about that a bit later. And then it kind of spiraled out from there. I'll stipulate, and maybe this isn't the best introduction for this kind of field and this kind of community, but I honestly didn't think the microbiology of the built environment was a particularly interesting topic. It's a mostly dead microbial ecosystem, necessarily so, right? We've tried to eradicate as much fungal growth, so molds and spores are bad for human health. We tried to get rid of all those, and we created a rather dry, cold, inhospitable ecosystem in these buildings. So for me, trying to explore this as a scientist, I was a little bit taken aback by the need to explore what microbes might be left surviving on the edge of life in this rather desperate desert of an environment. In fact, one of my graduate students, ex-graduate students now, an assistant professor, Sean Gibbons, has described this science as a desert ecosystem science. You know, like literally the built environment is a desert. And so, but it does present some rather interesting ideas, and hopefully you'll see from the work that I'll present that there is at least some decent research to be done if you look hard enough. So I'm also at the Center for Microbiome Innovation, led by Professor Rob Knight at University of California, San Diego. I'm the editor-in-chief for the American Society of Microbiology's journal M Systems, which publishes a lot of built environment research, and we encourage articles in that space. So the microbiology of the built environment is a really interesting space, right? But as I said before, the environment itself is pretty inhospitable. And it's predominantly designed that way. We try and reduce as much moisture. We try and keep it temperature controlled. We try and do everything to essentially make the environment comfortable for us, but it's not necessarily comfortable for microbes. And the predominant source of microbes in that environment, as our research and a lot of research has shown, is from the humans that occupy the space. In the majority of homes, offices, buildings, schools, hospitals, etc., the only living things other than microorganisms inside those buildings are people. And each one of them is emitting around 38 to 45 million bacterial cells every hour, mostly from exhalation and stuff coming out of our mouths, but from our skin, from our hair being released. And so it's a dynamic, vibrant ecosystem for that perspective. But when things leave our nice, warm, moist body, most of them die. So where does that leave our understanding of microbial systems dynamics? What can we learn by looking at the microbes that have survived in that environment? And in this review with one of my colleagues, Brent Stevens, who he's at the Illinois Institute of Technology, but prior to hooking up with me, really only explored how comfortable we could make buildings as a building scientist, not necessarily looked at the microbiology, but he's a convert like I am to this particularly niche area of science. And what he did was try and, what we did was try and here elucidate the types of mechanisms by which the microbiology in buildings might interact with our bodies and how that might influence our health. So, I mean, there's a huge amount of known research in here. The indoor air community and the indoor air conferences, which held in the United States and European Union regularly, are very specifically guided towards how can we manage indoor air quality to promote human health. But tagging microbiology onto that has been almost a niche area associated with just culturing. When we actually look at everything else, we can see there's a huge amount of microbial movement in these different environments, and I'll talk a bit about that later. But everything from bacteria and fungi and viruses, I should put SARS-CoV-2 on here, that we breathe in and interact with the immune cells in our respiratory tract and lungs, all the way through to things that we emit out as we breathe, which is a cornucopia of respiratory oral skin bacteria and viruses and fungi. And then fomite association. So when we touch surfaces, we pick things up. And then when we touch a surface, we also deposit microorganisms onto those surfaces. And it's very important that we can understand which of those interactions is important. And as I said, during a global pandemic, I'll talk about our SARS-CoV-2 research a bit later, which is very nascent and hopefully not too inappropriate. But in the context of this, obviously we know that touching surfaces leads to very, very, very few SARS-CoV-2 infections. That fomite transference is not really a route of transmission. The main and predominant route of transmission is airborne. And this is the reason we should all be wearing masks, no matter what the July 19th recommendations suggest. If you are taking public transport or inside a building or conglomerating outside with large groups of people in close contact, wear a mask. It's the only way to significantly reduce transmission. Microorganisms, when they finally find themselves in these dry, cold, inhospitable environments, predominantly go into a sense of dormancy until there's sufficient moisture around. And everything over the last 2,000 or 3,000 years, there's been a significant degree of literature, and I mean that 2,000 or 3,000 years. There's a significant degree of literature that talks about microbial growth, albeit only in the last 150 to 200 years has that really been focused on what we would call microbiology, including in the Bible, where it said if your home gets damp, you should wash the walls of your home with horse blood in order to try and eradicate the diseases which would occur. Basically, people were getting sick due to mold growth and the metabolites and spores that molds release, which have a significant amount of evidence associated with things like nasopharyngeal inflammation, cough, bronchitis, eczema, that atopic march that we associate with the development of asthma. So they suggested the only way to eradicate that was to freshly kill a horse and wipe the surfaces of your home with horse blood. You can continue that practice if you so wish. I would recommend maybe just trying to limit the amount of moisture that builds up in homes. However, despite the past 200 years of rather sophisticated developments in architecture, building practices still are leading to leaky pipes, condensation zones, hot and cold areas, which lead to moisture development. And when that happens, things grow. Most of the life on our planet cannot grow in the absence of water. In fact, I'd say all the life on our planet. And so you need that moisture. And when things start to grow, they start to produce metabolic output. And when the metabolites come out into the environment, that's when we start to see human health issues. And the chemistry developed by fungi and bacteria in these moist built environment spaces is quite complex. Everything from alcohols and amines and aromatics. And some of them, this is an interesting little adage and probably wrong, but it's still fun to say, some of them can have psychoactive effects. And so the idea that if you have a fungus growing on a wooden building and it's left alone for long enough, say a door is being closed 20 years ago, and the air builds up with their chemistry as the fungus is growing in this damp, moist wooden space. If someone was to finally go in and creak open that door that had long not been opened and walk into that environment and breathe in, they could have psychoactive responses that are associated with the chemistry from the fungi and bacteria in that environment. And some of those fungal psychoactive events may be leading to the ideology of ghosts. So, you know, who's to know that microbiology and ghosts could be linked? But we think that actually might be real. And, you know, and there's a lot of evidence supporting the idea that malaise or depression and certain mental health issues due to long exposure in buildings could be associated with indoor chemistry. And so there is active research in that area. We know that while the built environment is not a hotbed of microbial evolution necessarily, it is still a selective environment. The vast majority of microbial life in the buildings that we explore is dead, so it's not alive. The few things that are able to survive in the absence of water are usually spore formers, both the spores from fungi or the spores from bacteria, and they lay there dormant for a long time. But what's interesting is when there is a little bit of moisture, a little bit of oil around on those surfaces, we do see some microbial activity. And then we also see a very, very distinct selective gradient. And what these rather cryptic figures show is the balance between codon usage bias for genes associated with antibiotic resistance in bacterial genomes and the non-synonymous to synonymous ratio for nucleotides coding amino acids in those genes. And what this shows is that between, say, day 97 and day 170, there is a positive selection gradient for certain genes associated, interestingly, with antibiotic resistance, virulence, and survival. So heat shock proteins and other drought stress related proteins in those genomes, predominantly in Staphylococcus and Anerococcus. And this is work we published a few years ago. So here we took metagenomics and we sequenced it over time, as Darren was suggesting, and using that time sequence of metagenomic analysis, we can start to assemble those genotypes and examine how their genetic structure is changing over the course of that time. And there is a slight but significant selective pressure towards these virulence and resistance phenotypes. Interestingly, we also see this in space. So just for fun, we do work with NASA, and a paper we published a couple of years ago now, we took fungal isolates on the ground, which we'd isolated from buildings, and we grew them up, and we kept one of them on Earth and then sent one of them up into space to the International Space Station for a 3-month return flight. When that organism was grown on the International Space Station and then brought back down to Earth, we compared the ability of the fungi, the earthbound fungi and the flight-bound fungi, to kill nematodes in culture. And so you can see here, for example, that these F4 strains, which are flown strains, are significantly better at killing the nematodes than things like F3 and 293 GR ground flown, which have not been present in space. So we think there's a potential interaction there for fungi, built environment fungi, that might be relevant for space travel, and I'll talk a bit about that later. So exploring how microorganisms grow when water becomes available in the building is obviously paramount, right? It's great swabbing surfaces and seeing what's there, but with 99% of the cellular biomass or genetic biomass that you identify as dead or on the edge of survival, then it's potentially not very useful, right? Other than maybe in forensic applications, which I'll talk a bit about later. But here, we know that wetting causes growth. We know that when moisture is available, microorganisms will grow. So we took 3 different types of building material: gypsum, medium density fiberboard, mold-free gypsum. So this is gypsum with an antifungal agent in it, and then OSB, which is another type of board. And we either wetted them, and you can see these graphs here with these red and yellow containers are mold-free gypsum, medium density fiberboard, and OSB that have been wetted, or they remain unwetted, and these down here other than the gypsum. And we looked at the degree of fungal and bacterial growth on those surfaces. And indeed, when you wet something, stuff grows. Shock horror. It's amazing science. But what's interesting here is you can start to see, and of relevance to Darren's work, the relationship between bacteria and phage particles. So interesting, when there is more water around, we see more bacterial biomass, biologically active, alive, complete cells of bacteria. And when you have biologically active bacteria, you have more phage. So phage and bacteria go along side by side. Again, not massively enlightening, but still really interesting to see that we do see biological activity and ecosystem dynamic when there is moisture available on these built environment materials. And when we obviously see significant correlations between bacterial-like particles and the amount of moisture, so they go up when there's more water, same with the viral-like particles, hence the phage. And you can actually see differentiation between the materials. So here, for example, we see a significant degree of more bacterial-like particles of bacteria growing on mold-free gypsum. So when there isn't any fungi around, we see a significant increase in the degree of bacterial biomass. And this isn't a sequencing artifact or because this is actually looking under the microscope and doing staining assays. This is an artifact of the fact that in the absence of fungi, the bacteria can grow more because they're not being kept in check by antibacterial agents produced by fungi, and they're also not competing for the resources which the fungi need. And it's very interesting to see the talks coming up later today talking about that relationship between fungi and bacteria, because it really is central to the growth of microorganisms in built environments. And interestingly, bacterial and fungal diversity is very well correlated. We can look at that via some of these graphs, which do look a little complicated to start with. But here is the diversity, the richness of fungi on the y-axis, and on the x-axis is the richness of bacterial types. And we see that when we have more bacterial species, we often have more fungal species. Interestingly, Bacillus and Pseudomonas growth, the increase in the proportional abundance of Bacillus and Pseudomonas across these surfaces over time is significantly anti-correlated. So we actually start to see that if you're growing Bacillus, then Pseudomonas can't grow, and if you're growing Pseudomonas, then Bacillus can't grow. Right? There's a relationship there between those two groups. Here you can see in red Pseudomonas and in blue Bacillus growth on wetted substrates with the dotted lines. But interestingly, Eurotium grows very well without water. So you see Eurotium here in red, a solid red line being non-wetted surfaces. And we actually see Eurotium growing. Eurotium is actually very good at sucking water, moisture out of the air. So if you have a high humidity environment, anything over about 70% relative humidity, then Eurotium will be able to grow on those surfaces. Penicillium is not so good at doing that, but when there is moisture around, as this dotted blue line here compared to the red dotted line of Eurotium, then Penicillium can grow very, very well. So it actually outcompetes Eurotium. So there you have niche structure depending upon the availability of water. If we look at the chemistry using targeted and untargeted metabolomics of these same surface substrates, and again, remember the x-axis is days of exposure. I've gone up to 6 days and 8 days for some of these. And then the y-axis is the concentration of those metabolites. And then we have gypsum, medium density fiberboard, mold-free gypsum, and OSB. The top is metabolites and the bottom row is microbes. And you can see that antibacterial alkaloids like nigrogillin and fumiclavin C are correlated with the abundance of Ascomycota and negatively correlated with the abundance of Bacillus and Pseudomonas. So if you look at gypsum here, for example, see a big spike in fumiclavin C, and you see here a significant increase in the amount of Bacillus and a significant increase in things like Penicillium, which is growing as the Ascomycotal fungal group in the presence of that community. And things like Eurotium aren't growing very well there because it's not enough, it's too much moisture around. If you look at mold-free gypsum, you see little spikes of fumiclavin C and nigrogillin, which are associated with the community, but that's only when Eurotium has a little burst of growth. Otherwise, Bacillus and is growing very strongly in this community. Interestingly, the concentrations in Nigulin and Fumicamin C are significantly anticorrelated with Bacillus and Pseudomonas, so we think that the fungi are producing these in order to knock down those bacteria. And if we do autocorrelation indices using either a Bayesian math to look at conditional codependencies, or in this case, just using correlative co-prediction, so artificial intelligence or machine learning based assays to identify correlative codependencies within the community dynamic, we can see that things like Bacillus here in yellow, down here in yellow, and Pseudomonas viridiflava here in purple are significantly anticorrelated for certain types of metabolites. So things like atoxystrobin are very positively correlated with the growth of Bacillus and negatively correlated with Pseudomonas, whereas scopoletin is positively correlated with Pseudomonas and negatively correlated with Bacillus. So we think this is bacterial warfare. As these organisms grow, they produce chemistry in order to try and eradicate other organisms. And when we look at Bacillus, which the vast majority of built environment associated bacilli are not particularly dangerous to human beings, and we think we could leverage this for bioprotection in environments such as buildings, and I'll talk a little bit about that later. So back in around 2012, 2013, we got a sizable grant to examine what happens in a building that becomes operational. So we already knew that human beings are the predominant source of bacteria into these buildings. We wanted to really look in depth over time and a very, very dense time series to examine what happens when a building first becomes operational. And the best place to do that is a hospital. So this is the Center for Care and Discovery at the University of Chicago, where I was at the time. And here we were able to examine these top floors, these 3 top floors, which are the patient rooms and staff quarters and nursing stations, and examine what happens in this building before it becomes a living, working hospital, when it's being super sterilized to make sure that the patients can move in. And then what happens when the staff and the patients move in. So we did 365 consecutive days of monitoring. Let's check the chat. 365 consecutive days of monitoring, 2 months pre-opening, 10 months post-opening. We examined things like the floors, the bed rails, faucet handles, the air and the water, surfaces in the nurse station. And then when the patients and the hospital staff arrived, we also continued to analyze those. We produced over 17,500 samples, of which around 8,000 or 9,000 samples produced enough biomass for analysis, and then enabled us to significantly examine microbial dynamics in the space. If we look here on the left, I have a principal coordinate plot. So every single dot here is a microbial ecosystem, and the closer they are together, the more similar they are in composition and structure, and the further away they are, the more different they are. These are all floor samples, so room floors in circles and station floors in triangles. You probably can't see the difference. It doesn't really matter. The blue circles and triangles are all pre-opening. So before the hospital became an operational structure, and then flipping to the day after it became operational, when the patients and the staff moved in, which was literally over a 24-hour period, we see a significant shift to the community structure into one that looks very different. And that's really dominated by an increase in the abundance on the floor of human-associated bacteria. So, for example, Acinetobacter becomes less abundant on the floors, but Corynebacteria, Staphylococcus, and Streptococcus become significantly more proportionally abundant on the floors. So we see a switch from a building where you've got a few bugs on the edge of survival, potentially associated building materials, to one that's rich in bacteria that are associated with human skin or respiratory tracts, gastrointestinal tracts, etc. Interestingly, we can examine this because we have this very dense time series, daily sampling, metagenomics, 16S, ITS, various omics thrown at this analysis. We're able to examine this entire structure over time. And what we have here on the y-axis is the distance between the microbiome of different staff members. And because this is a dissimilarity matrix, as the number goes higher, the samples on people's skin are more dissimilar. And as the number gets lower, the samples are more similar. And so if you get here, this is actually the summer during the humid months. We actually see a significant relationship between humidity and the microbial similarity. And as you get into the humid months, this is in Chicago, where we get very, very high humidity and very high temperatures, 35 to 40 degrees centigrade, 80, 90, 95% humidity. It's incredible. And it invades the building, no matter how much building control you have. We start to see that people's skin bacteria, this is of the staff, tend to look significantly more similar than they do during the very cold, dry environments that occur during the winter. So this is a significant seasonal relationship. It's very important that you have a very dense time series if you're going to analyze these environments, because you want to capture what's going on at different times of the year. I now live in Encinitas, where it's usually 21 degrees centigrade every single day and sunny. I'm now in London, where it's currently sunny, but it was raining this morning. It'll be raining at lunchtime, and it might be sunny a bit in the afternoon. It'll be raining again. The kind of climatic variation is crazy if you want to really understand what's going on in different environments. We can also track, using Bayesian statistics, the significant movement of microorganisms between sites within a person and between people. So this is a good example of that. On the left, I have a staff member. That's their hand there in the middle, their pager, their shirt hem, their nose, cell phone, shoes, gloves, etc. And then on the right, I have a patient. Their axilla is their armpit. And then they have the bed rail, their hand, and their nose. And what you can see here predominantly is that staff are a constant source of bacteria to the patient. So, for example, the staff hand shows a significant relationship or movement of staff-associated Staphylococcus and Corynebacteria to the armpit of the patient. And interestingly, Acinetobacteria to the patient's hand. But the patient is never really a source of bacteria to the staff. That doesn't necessarily make sense, right? Because if you're interacting with someone, you're going to be microbially interacting as well. And I'll mention that a bit later. But what this really does indicate is the power of hand washing. So when the staff member interacts with the patient and they see that microbial swapping, as soon as they leave the patient room, they have to wash their hands before they see the next patient. And in doing that, they eradicate that microbial signature they picked up from physical interaction with the person. And as long as that physical interaction isn't too extensive, that's one good way of reducing the spread of disease. It's remarkable how this proves the importance of hand washing, and yet people still question it. That I remember I mentioned at the beginning, we got a $125,000 award to examine microbial communities in people's homes. And I've long been a proponent for the last 15, 20 years of time series analysis. I think time is the most important thing if you're going to analyze any ecosystem structure. And so what we did is we did a daily sampling of a number of different homes over a period of 6 weeks. So we did this very dense time series over a very long time period that allowed us to really understand how microbes might be moving. And lots of information in that paper. You can look it up, 2014. But some of the fun things are that we can map the movement of different bacterial groups between different people. And I just colored them. I haven't colored the movement. I've just showed all the different types of interactions. Feet tend to have more microbial interaction with floors than they do with surfaces that hands interact with, and vice versa. Hands are interacting mostly with those top surfaces. You can see a lot of interaction between hands and noses, for example, even hands and feet to a certain extent. And interestingly, this is a community with 2 adults and 2 children, and so the hands of the adults are nearly always more interacting with the feet and the hands of the children than the hands of the children are with the feet and hands of the parents. So you can see that the parents are looking after the kids. Interestingly, when we add dogs into this kind of situation, we see a significant disruption of that microbial paradigm of just a family with people with humans in it. Here we have a family with furry pets as well, and the dogs significantly increase the movement of microorganisms, both within the home, but also between the people and between the surfaces of the home. And in parallel to this study, my colleague Rob Knight also did another study. We looked at 160 couples, so twice that number of people, and couples with a dog in the home shared more microbial similarity than couples without a dog. Interestingly, couples with a newborn baby shared less microbial similarity than couples that owned a dog. So it looks like dogs could be potentially an incredible superhighway for allowing the movement of microorganisms between people, probably because they're so promiscuous, right? They want love from everybody, and so they spend a lot of time roaming around and interacting with people and increasing that degree of microbial sharing between people. My wife, when she read that paper and the press release, made me rescue a dog. So this is Captain Bo Diggly on the beach in Sanitas, and I've been proposing that dogs are awesome for a long time, and I even trying to get NASA to allow me to put dogs on spacecraft, because I think it would just be super cool. The first dog went up into space and died there. Well, let's have living dogs in space. That would be cool. Obviously in spacecraft, not just floating around, because that would be weird. As a short step to that, NASA has given us a grant to explore what the time dynamics of microorganisms look like on spacecraft. So here we're looking at antibacterial resistance and fungal resistance and virulence on the surfaces of the International Space Station. And in the context of this study, we are surveying different surfaces with techniques that keep the microorganisms, bacteria and fungi alive, and then bringing them back down to Earth, culturing them, doing genome sequencing, and comparing it to metagenomics of preserved cells, so that we can really start to understand what's living on those surfaces, what's viable, like can be culturable, and what's their antibacterial resistance and fungal virulence. And actually, interestingly, this is ongoing. So our first SpaceX flight went up on June 3rd. This is Megan MacArthur, who's actually an alum of the Cripps Institution of Oceanography, where we work, and she's now an astronaut. And she is our main sampler on board the ISS. This is her sampling on Friday, and you can see in her glove she's got one of our wipes, and she's wiping one of the panels on the ISS right there. And collecting those microorganisms. That keeps them alive. We keep them at 4 degrees, and we bring them back down to Earth at 4 degrees, ship them to our labs at 4 degrees, and then we grow up what we can. And we also have samples that are stored in freezing temperatures in order to do metagenomics on living cells, which we use PMA for anyone asks. So we try and keep only cells that are alive and get rid of any DNA that is extracellular or dead cells. We've shown for a long time that microorganisms can grow quite significantly in these environments. Let's check my chat. Sorry. Oh, perfect. Questions coming. I'll do those later. People do physically interact with the environment, and here, back to this home study paper that we had a couple that lived with their lodger in the same home, and the couples, all the man and woman living in red and blue here, are significantly more similar to each other than they are to the lodger. There is some overlapping similarity, but on the whole, they are significantly different. You can see that hands are significantly more similar between the couple. Their feet are significantly more similar than they are to the lodger. Interestingly, their noses are all very different. The nasal respiratory microbiome, as I'll talk about a bit about later in reference to SARS-CoV-2, is always very significantly different. This provides us with an interesting narrative to tell. To take this to its obvious conclusion, we also explored the microbial dynamics of people in a new college student dorm rooms. These are first-year college students. So we got in there and we looked at the interactions. So this is again one of our Bayesian dynamic interaction diagrams, and it looks like a complete mess. But the take-home message is that we can track the movement of microorganisms predominantly through hand-to-hand, hand-to-surface transmission, but also through respiratory transmission in the nose. And interestingly, from this, which we can't really publish, we could tell exactly which students were interacting with which other students in which rooms, which, as you can imagine, for the parents of those first-year undergraduates was a little bit concerning. So it wasn't published in the paper, obviously privacy reasons as well. But it's interesting to note that you can use this information to kind of track activity, which brings us on to the idea of microbial forensics. Just for fun to start with, these are different surfaces in a home. The x-axis is time, and the y-axis is the percentage of bacteria found on that counter which originated from one of 6 occupants. There's 3 humans and 3 dogs. Person 1 in red is a young man, 25 years old. He lives with his parents. His father is person 2 in orange, and his mother is person 3 in yellow. And immediately on the kitchen counter, you can see the horrendous gender imbalance in this home, as often 100% of the bacteria that we encounter on the kitchen counter originated from person 3, the mother, until she goes away for a couple of days here. Person one went away here, and then the mother went away here. And then the father has to desperately fend for himself in the kitchen as the mother's no longer there, probably desperately trying to make toast, to disparage the male gender there. On the floors are predominantly dog bacteria, apart from this weird blip on the bedroom floor for person one, which I don't like to talk about. And on the doorknobs and light switches, you have dog bacteria, not because the dogs are reaching up and touching them, but because you have that fomite transference from interacting physically with a dog and then touching one of those surfaces with your hand. So you act as a transmitter of dog bacteria to different surfaces. What's interesting about this is we can obviously track what people are doing in that space. And so we got a couple of grants from the National Institutes of Justice. That's the Department of Justice's research wing at the US federal agency. And what they allowed us to do was go into different homes and create mock burglaries. So we broke into homes. I don't have the slides here, but it's an interesting story. We broke into homes and then we went in afterwards and we swabbed all the surfaces and we said, can we identify a burglar that broke into this home? So we obviously had the burglar in our database, their skin microbiome, their oral microbiome, their respiratory microbiome. And so could we detect that person? And invariably we could. So if we knew exactly which person broke into the room, we could see their bacterial genotypes present in the home. So they did shed bacteria when they were present in the home, interestingly, even if they're wearing gloves, right? And when they in the home, and we found them there, or the office, we did an office as well. We found those bacteria. We found which people they were actually invading that space. What's really annoying though, is if you actually compare that person and their signature in the home to over 1,000 people and their microbiota, then your ability to detect the individual disappears completely. It's statistically improbable that you can identify that person as having invaded that home beyond a reasonable doubt. And so I like to think of this as Hercule Poirot, Agatha Christie style microbial forensics. If there are 6 people in a room and I know one of them did it, I can tell you which one it was based on the microbial signature they left behind. But in a court of law, in the real world, it's technically impossible because the degree of microbial identification, even at the genotype level, leads to too many inconsistencies at the statistical level. And we did publish that in the International Journal of Forensics because we wanted the legal teams that might try and use this science to read that paper, and we promoted it widely in legal circles internationally. So just to finish up, because I know we've got a few more minutes, I want to talk about the stuff that we did with SARS-CoV-2 because, hey, it's a hot topic and it's a pandemic. What we originally worked on with the CDC was trying to identify how long SARS-CoV-2 might remain viable on different surfaces. And so this early paper from Dora Milan came out in the beginning of 2020 and demonstrated that, yes, in aerosols, it survives quite some time. On copper, it dies off very quickly. On cardboard, it can live for about 24 hours, stainless steel up to 70 hours, and plastics up to 80 hours before it's extinct. And that was an interesting phenomenon, right? And we now know, as I've said before, that SARS-CoV-2 doesn't really transmit from surface to surface. 95% of all the transmission events that we see occur via respiratory inhalation, and therefore that's the reason masks are so important. But we've always considered it interesting to know what might be going on with that surface to surface transmission. And so we wanted to know, because I'm a bacteriologist, not a virologist like Darren, I wanted to know what the bacteria could be doing. And so we teamed up with a colleague of mine at the St. Jude's Hospital in Memphis, Tennessee, who was trying to examine how bacteria might be involved in the transmission of flu. So in this work, he was exploring how flu virus can bind to the outside of different bacteria. And the hypothesis being based on in vivo evidence from ferret models, which are very good for respiratory tract analysis, we were able to demonstrate that the bacteria can actually be more effective at infecting human cells in the respiratory tract if they're covered in flu virus. So, it's like the flu virus has a particular lock or a key that can break into the cell's locks. The bacteria doesn't have that, but if it coats itself in flu virus particles, it can easily break into the human cell lining the respiratory tract and cause infection more readily. So it's like a cloak that allows it to break into the body. Interesting though, if a flu virus is bound to a bacteria, it can actually survive longer outside the body. So there is a two-way street here. And in this graph here, we see the presence of bacteria without any desiccation. We can see that we have a lot of flu virus particles that are active. If there are no bacteria absent and we have desiccation, as you would have on any surface in a building, the flu virus is virtually eradicated immediately. It doesn't survive outside the body. But interestingly, if you have bacteria and desiccation, then the flu virus can survive outside the body for more than 48 hours. I think my colleague has taken this out to about 7 or 8 days. So there is a predominant length of time which the flu virus can survive on inhospitable surfaces around the building because it's bound to bacteria, right? So bacteria can be involved in viral stability. So we wanted to know if the spike protein of SARS-CoV-2 showed any associated affiliation with cells of different bacteria. So here we took things like Streptococcus pneumoniae, Staph aureus, Pseudomonas aeruginosa, Haemophilus influenzae, and we bound a modified conjugate of the SARS-CoV-2 spike to the outside of these cells and saw if it bound predominantly, and indeed it did. So A is the actual microscopy image with calorimetric identification, and B, we've made it pretty, right? So you can see the cell and you can see SARS-CoV-2 spike protein binding to the outside of the cell line. So that's really interesting, right? The bacteria does seem to bind spike. So SARS-CoV-2 can theoretically bind to the outside of different bacteria. So we wanted to examine this in different surfaces on different hospitals. So back in early March, prior to the first positive SARS-CoV-2 cases being brought into the United States off of the Princess Queen of the Ocean or whatever it was, the cruise liner, who brought those patients into our hospital, we were able to sequence the surfaces and the staff members of the hospital prior to any SARS-CoV-2 cases being brought in, and then examine it over time. So as soon as a patient arrived, we start swabbing the patient and the building and the care workers. We do it on day 1, day 2, day 3, until a patient discharge or terminal cleaning associated with SARS-CoV-2 death. And so we were able to examine this very dense time series of all of the SARS-CoV-2 cases that came into our ICU and examine them with context of the built environment. And we had a dedicated team of people doing this. This is just some of them. Dr. Eklu Mensah is one of my wonderful postdocs, Dr. Maratz, again, one of my wonderful postdocs, and Dr. Pedro Belda Ferrer, another postdoc, and then Neil Gottel, Shoka Dera, and Sonia, who are graduate students and technicians, who spent all of their time through March, April, May, June, July 2020, working in the hospital, working in the lab, and selflessly giving of their time during a pandemic to really start to understand this as quickly as possible. We're able to track SARS-CoV-2 in patient rooms, even in non-ICU rooms where we had SARS-CoV-2 patients in recovery or in early stage prior to ICU admittance, and then in ICU rooms. And the surface which had the vast majority of SARS-CoV-2 positivity were floors. So again, next to the bed was always worse, highest number of SARS-CoV-2 positive samples, but you find it on the floor in the room outside of the room. So a staff member comes in, they're interacting, they leave the room, they're tracking SARS-CoV-2 particles on their shoes. And some of these, the viability was still present there. So in the patient, viability and copy number was significantly elevated for up to 27 days after onset. But we even find it on the hospital surfaces at very low densities, but it's still viable and medium density, so 10 to the 3, even up to 2 weeks after the patient was admitted to the room. So it is surviving and it is stable. Interestingly, using a patient microbiome, we can predict that the patient has a SARS-CoV-2 positive case. If you look at the stool and the forehead, it's okay. You get reasonable prediction, 0.68, 0.75, but obviously the nostril microbiome has a significant ability to predict SARS-CoV-2 positivity. And interestingly, if we take all of our surface samples, so floors, bed rails, everything, right, and the surfaces inside the patient, and we say, okay, which microorganisms are best involved in predicting whether a patient was actually had a SARS-CoV-2 positive case, then the one microorganism which had the best statistical probability of predicting it, no matter what environment it was in, is Rothia denticorosa. And you can see that here, that, on the stool, the forehead, and the nares, it's highly significantly correlated with the abundance, with the positivity of SARS-CoV-2. This is interesting, right? Why Rothia denticorosa? Well, this data is only about a week and a half old, but we were able to demonstrate that Rothia denticorosa does bind spike, and it binds it better than any other respiratory bacteria or oral bacteria that we previously identified. So staph, strep, Pseudomonas have nothing on Rothia denticorosa. It appears to be binding onto the spike protein and binding onto SARS-CoV-2 conjugates, which I haven't shown here, but we've just demonstrated with our colleagues, and sticking for a long time. And we were able to demonstrate by comparing our data from our hospital study with another paper, which was published in 2016, that Rothia denticorosa and disease state is not just associated with people in the ICU. It's not just found more abundant in sick people. It's found more abundant, or more proportionally abundant and physically abundant in people with SARS-CoV-2. So it's really interesting, suggesting that Rothia denticorosa and certain other organisms could be playing a significant role in not only the pathogenesis and disease of SARS-CoV-2 sickness in the ICU, but potentially also in transmission by binding that SARS-CoV-2 virus and allowing it to survive outside on the floors and on surfaces outside the building for a longer time. Interestingly, despite the long-term shedding of patients, and I was finding all of this, no healthcare workers in our ICU came up as SARS-CoV-2 positive or were hospitalized associated with SARS-CoV-2 sickness throughout the analysis. And these are our wonderful doctors, our chief of the ICU, who was giving us full access, and one of my wonderful clinical fellows, Farhana Ali, who gave of her time to work with the patients through some very, very trying times. So she did an amazing job. So patients can shed virus for several weeks. Viral stability on surfaces is influenced by bacterial biomass. The standard precautions are adequate for preventing transmission from surfaces and patients to healthcare workers. And there seems to be a relationship between SARS-CoV-2 and certain bacteria like Rothia, both in the body and on surfaces. But those mechanisms are still being discovered, and we're still trying to desperately hunt down funding to really dig into it. In the meantime, spending money like crazy trying to do it without funding. So it's a cutting edge Wild West style research area, but it's fun. I want to say thank you to some of the funding agencies that funded the work I presented here. And some others down here. We work with a lot of different agencies and a lot of different organizations, companies, and individuals to get our research, and we're always grateful. And to my wonderful research team, this is us on the beach just pre-pandemic. This is March 6th, and they worked tirelessly throughout the pandemic, and we are continually exploring how to expand our research team. So please let us know. And I do have a book out which I have to advertise each time. It came out in 2018. It's about human immunology and microbes. So if you have a kid, you might want to buy that. Shameless self-promotion time. Thank you very much for listening.
Thanks, Jack. Amazing talk. I mean, we have some questions in the chat, but I've got a couple of questions myself first. Especially regarding the SARS project, really interesting stuff regarding the interaction with the spike and the bacterial surface. If you titrate the recombinant protein and you crank it up, do you see complete surface coverage, or is it site specific on the cell surface?
It's interesting. There is a density-based relationship. We're still exploring to what extent we can manage that density, but it does saturate. Not the whole surface, it saturates at a particular concentration. So it looks like there's a conformation which is allowed, but it isn't total surface saturation. Really interesting. I mean, I think we've got quite a lot of untargeted sequencing, from in swab samples where we've not done PCR. It's basically just been complete metagenome side of things. So I wonder if, and it's RNA based, so I wonder if we can see transcript from some of these bacteria in those samples. It would be well worth to go and have a look.
So if we go through to the chat, what's there? Oh, so one of the other questions is, how much horse blood do you need to sterilize a wolf?
I think the Bible says you have to kill a whole horse, so
Whole horse.
probably about 15 liters, pints or
That's
so. I can't remember how much is in a horse.
So the first cut of the question there was regarding the Eurotium and how it was able to survive the dry conditions compared to the others.
Yeah, we don't know. So there's been a fair amount of work on this in the past by other indoor scientists. So it looks like it promotes condensation from moisture in the air. However, the mechanism, as far as I'm aware, is not well established. But it does seem pretty good at being able to forage moisture from the atmosphere in very dry conditions.
So when you were wetting these surfaces, you have different surfaces, probably different porosities and absorbance levels. Did you see any correlation between that level of absorbance and just was there like a greater, was there greater movement in certain species because of that?
Yeah. So what remember, all of these are they're fiberboards, right? They're things which the average American home is built out of. I say all American homes are, unlike all British homes, are virtually never made out of brick or stone. They're all made out of wood with fiberboards for the walls, and so most of those do have a certain degree of porosity and absorbency. What we didn't do in this study was do plastics, glass, metals, and stones, which would have potentially shown a significant difference, but there was no significant difference. The only significant difference in between absorbency and moisture content of the material was in the mold-free gypsum, which was eradicating the fungi. So it had nothing to do with the absorbency ratio. And we think that's because there wasn't a significant difference between these different substrates.
Yeah. I think the second question there is regarding the hospital study and post-opening. They said they were surprised to see no enterics found post-opening in the hospital. Because they found it quite, as persistent colonizers in hospitals. And they didn't ask, is there any viability assessment done then alongside that?
No, enterics. Yeah, so they're there, but they're very, very low in abundance. And they're quite variable. So it's not like you get a lot of Enterobacter. It's not like you get a lot of, and bear in mind, we're in the hospital room, not in the ensuite toilets, right? So if you go into the toilet, you start to see more enterics there, but most of those enterics can't survive for very long, right? They die. This is why I don't necessarily have an issue with flushing the toilet in the bathroom. Yes, you're going to get a plume of organisms, but it's not really going to harm you unless you've got a massive enteric infection and somebody else is sitting there right next to it breathing it in. For the vast majority of the time, it will go up into the air, it will separate down, and those things will die. They don't survive for a very long period of time on those surfaces. I'd be interested to know if there is any data. You said any viability assessments done, then yes, we did culturing of the organisms at the time. That didn't make it into the paper because the reviewers asked to take it out because it was sporadic. It was done by the hospital themselves, but we didn't find any viable enteric organisms in the hospital room and on the counter. We only found things like staph and strep, which do have that ability to survive for slightly longer than the enterics.
And then the next question in the chat is regarding was any longitudinal sampling done with the couple living with the lodger? Wonder if the difference is due to a close contact or simply length of time living in the same building.
Yeah, that's 6 weeks of daily sampling in that couple with the lodger as well. And yeah, I mean, what we haven't been able to do yet is take a couple who have never really physically interacted before and follow them over the duration of their relationship, because of the amount of time it would take and the lack of interest. We need to do it in probably 20 or 30 couples, and the number of samples would be phenomenal. But the hypothesis, your physical interaction, the length of time you're actually interacting for are going to be important, right? But remember, this couple and this lodger were living in that space prior to us coming in and sampling it. Yeah. And then we sampled it every day for 6 weeks, and that trend was maintained over that period of time. We can that kind of temporal analysis is in the paper.
There's a question there that says, regards to the microbial forensics, how transient are the individual microbial signatures? Does each person have a lasting bacterial fingerprints, or does it get muddied the water if they shower? So kids have got a constant one.
Yeah, that's true. If you don't go into you don't wash like my kids don't, then yeah, you could have a constant one. I mean, yeah. Remember, we did this stupid study for a radio show in New York where we took Neil deGrasse Tyson, who's a physicist and director of the Hayden Planetarium, and we had him shake hands with a reporter, and then we swabbed their hands every 10 seconds after the handshake. And we did 10, for about a minute before, and then we did it for about 6 minutes afterwards. And you could see that the signature was present there. If they washed their hands, then the signature disappeared, and the only signature was left was the signature of their own skin, right? So I like to think of skin as like a city, right? It's like, think of Manhattan, and at the top you've just got the top of the skyscrapers, but the microbes exist really deep down there into the sebaceous oil ducts and the crypts and the valleys and the cell lines, and deep down in the cell dermis, there's anaerobic activity going on. There's things like short chain fatty acids being produced by fermentation of sugars. It's crazy stuff, right? In the skin, but that's really deep down. If you wash your hands, you eradicate whatever you just touched and whatever's bubbled up to the surface. But then 30 seconds later, the skin microbiome has bubbled back up to the surface, because it's quite a dynamic environment. It's not static as we think it is. And so, there's you can eradicate the signature of the other person, but your signature is going to be present there. So a burglar going in, if they wash their hands, they're still going to have that signature on there. Remember, our burglars were wearing gloves. And so everything was coming out of their nose. And so you can gargle as much alcohol as you want, but you're still not going to eradicate everything in your respiratory tract and your oral cavity. It's just not how it works.
Yeah.
So, I think that's important. But again, you got to go down the Agatha Christie route, right? These things are stable for up to about 15 to 20 hours based on our analysis. So we had someone touch a surface, and then we sampled it over a longer period of time, and we start to see the signature disappearing in about a day. So someone would have to get in there quick and know that 1 of 5 people did it. So it's a bit of a crap forensic tool, really.
It does sound like the next generation of parties with a midnight, doesn't it? Really.
Exactly.
Yeah. So there's one last question. I've got a couple more questions, but it's within the interest of time. But there's one there regarding shedding and the amount of clothing the person is wearing. Can it be avoided by covering the bodies? I think basically somebody's trying to work out if they can get away with it. I think is
yeah, potentially. I mean, you'd have to go in there with a very, very high degree of non-porous clothing, right? I like to say, and we've done this test, if I swab all of the chairs of an auditorium, I can tell which person was sitting on which chair, because your trousers are like loose fishnet stockings to bacteria, right? And so your gastrointestinal microbiota or your urogenital microbiota are left behind on the chair you're sitting on. That's just biology, right? The weave on this is huge compared to microorganisms, and so it doesn't make a lot of difference.
I think in the interest of time, Jack, it was such an amazing session. Thanks for all the questions, everybody. Virtual clap. I don't know how you do that these days. Many thanks, and we look forward to you staying around for the rest of the day and joining in when you can.
Absolutely. Thank you, Darren.
Thanks,
Appreciate
Jack.
it.
Take care. Bye-bye.
Cheers.